Ladies and gentlemen, I am Alain Tressot, the Vice President of the Academy. And for me, it's really a great pleasure to be here again for the second time. And I want to thank the President of the Academia das Ciências de Lisboa for giving us this great honor, and also Professor Armando Pombeiro for doing so much work during this meeting. So at that time, this was 2017, I was the president of the Academy, and Professor Martin was the great organizer of the meeting and also kind of vice president. So you see, 7 years later, things have switched, but both of us, and especially President Martin, has the strong will and wish to improve our academy on different points, to have excellent fellows in the academy, to improve the relationship within Europe, Western, Central, but also Eastern Europe, which is very important, and also to improve relationship with countries all over the world. You spoke of the relationship between South America and Europe, which should be developed, and we started to improve those relations with East Asia, in particular China, with a lot of success, I would say. So now I start with some emotion and honor to this session for the 3 introductory lectures given by famous scientists. And the first one will be given by Professor Elena Nader, was the head of the Institute of Pharmacology and Molecular Biology of Sao Paulo, and since 2022 became the first woman to be president of the Brazilian Academy of Science. Professor Nader has been a model for many women in Brazilian university, helping to promote the empowerment of women and breaking barriers in society. Her contribution go beyond research, having contributed with initiative aiming at diminishing the gap in scientific development between the different regions of Brazil and improving nationwide levels of high school education. So it is a very important personality for the world science, and Professor Nader will give a lecture on navigating the complexities, challenge facing health and social welfare at modern world. Please, this is Nader.
Good morning. For me, it's an honor to be here, and I want to initiate by thanking this beautiful place to host all of us in the same attitude, science for sustainability, and as pointed out already by Professor Pombeiro, Professor José Luiz Cardoso, and Professor, our president from EurASc, sorry, and leave no one behind. If we pay attention to that, we're going to change the world. I still hope that we'll be able to do that. So, in here, right? Initially it was said that Brazil, the Brazilian Academy of Science hosted the Science G20 group. And the topic that we had was Science for Global Transformation. And we have 5 different aspects that were covered. One was artificial intelligence, the second one was bioeconomy, The third one was the process of energy transition, health, especially looking at equity and social transformation that indeed percolated all the other four. So I decided to bring not the topics that we discussed, but some of the aspects that came out on those discussions between the 19 countries plus European Union and African Union. So briefly today, what I'm going to do is discuss why this topic, the demographic shifts, the impact of climate change on health and social welfare, mental health crisis, I think we need to pay attention to that, the technological advancements and the conclusions. So the challenges are interconnected and face strong impact with the demographic shifts, technological advancements, and the developing nature of global problems, such as wars and population migration, as already pointed out by the president of the Academia de Ciências de Lisboa. The complexities of these systems arise from intricate balance between resource, equity, and efficiency, and they are and can be exacerbated by climate change, social inequalities, and within and among countries, because we have inequalities within each country, besides inequalities among globally speaking. And we all face demographic global population change in a different way for each country. We're going to check that. And this challenge calls for adaptive policy, as already mentioned, effective governance, and innovative solutions. The 17 goals, in my opinion, health and social welfare are present in the 17 ones. It's not only because if you go to food, security is health. If you go to water, is health. So we have health in all of them. And of course, the 17 SDGs. And we have a very short period to achieve the goals. 2030 is tomorrow. Don't forget that. And, whoops, too fast. Back, please. Oh, too fast again. My goodness. Well, this is a paper that it's very interesting. It was published on Nature by Jeffrey Sachs and a very known personality, Mariana Mazzucato, who is an economist. And they took the 17 SDGs and divided in a different way. And they call Leave No One Behind, Science for Sustainability. So they transformed that in 6 transformation groups. So transformation 1, education, gender, and inequality. Health, well-being, and demography. Energy decarbonization and sustainable industry. Number 4, sustainable food, land, water, and oceans. Number 5, sustainable cities and communities. And number 6, digital revolution for sustainable development. And all of that are in the 17 SDGs. Demographic shifts. If we look at the globally, is the left panel, upper, you can see that, and in the next one on the right side is Europe, you can see that's totally different. So there is no way of comparing one with the other. And approaches are going to be totally different, globally speaking, and for each country. So for Europe, the peak relating to getting older is already there. But globally speaking, it's going to take longer. And that includes countries that have more than 2 billion people, and you're going to see some of the slides that they're still with a young generation. So the approach for equity in Europe is going to be different than the approach for some countries in Asia, for instance. North America is very similar, and Latin America. We are almost equal to what's going on in Europe. A little bit delayed, but not that much. And the next one, you can see Eastern and Southeastern Asia. And then on the other one is South Asia. You can see that the peak's totally different. And Southern Africa and Africa as a whole. So each country needs to pay attention in the way they're going to behave to achieve equity. And do not forget, artificial intelligence is going to have a huge impact on equity if we do not pay attention to that. It's going, in my modest opinion, it's going to increase the inequalities among countries and within the country if we are not prepared for the young generation and poverty. Next in here. So the demographic shifts, the implications are there, and it's going to impact societies and their economies. The trend is aging population, especially in developed countries. Countries need to consider the profile of their population. As life expectancy increases, healthcare systems are strained by the growing number of elderly individuals, like myself, requiring long-term care, chronic disease management, and specialized geriatric service, which is going to be totally different if we go to some countries in Asia, for instance. Countries need to anticipate the size and age distribution of the workforce that will pose economic challenge and shrinking of the working force. And this will have an impact on social security, pension systems, and welfare programs. So governments, in my opinion, need to restructure, start to think how they're going to restructure pension systems, healthcare service, and social welfare in order to ensure that younger generations are not going to be disproportionately burdened. It's a task tough. Here's a paper that was published about 2 years ago, and they are showing the panel A, the percentage of population according to age, and panel B, the types, how many different chronic disease, 0, 1, or 2. And you can see that, as expected, it increases with age. And here is regarding the impacts of climate change on human and planetary health. So air pollution, change of in-vector ecology, and goes on and on. This is a beautiful map that was performed by CDC from USA. And of course, it's going to impact on health, cardiovascular disease that were not common in Europe, for instance, schistosomas or dengue. It's coming all over because of the climate change. And we need to be aware of that in order to provide the good health system that everybody would love to have. And the rising in temperature is going to influence environmental degradation and is going to have a terrible impact on public health and social welfare. Extreme weather events like heat waves, floods, and wildfires can lead to direct health impacts, including injury, heat-related illness, and respiratory problems. And this is going to impact food security, water quality, and spread of infectious disease. And this will have an impact on health system. Vulnerable populations, such as those on low-income regions or coastal areas, will be disproportionately affected and will have an increase in inequalities. Here is I brought some pictures to illustrate. This was in the south of Brazil, beginning of this year, 2024. The state of Rio Grande do Sul, 25 cities were totally destroyed. And the amount of water that came down was totally unexpected, similar to some places in other places of the globe. On the other hand, this is Rio Negro, the Black River, the Niger River in the Amazon, totally dry. You can see the main, let's see where it's in here. Well, here. This used to be the river. And now you can cross by car. And here is the typhoon that a few months ago destroyed part of Vietnam. And in here, oops, I want to show that because that's very important here. Here's a map showing the smoke. Remember the burning in Canada? The smoke went down to USA, but also moved towards Europe. And that's a map showing Portugal, Spain, and so on. So globally, we are interconnected. And climate change is a reality, and we need to pay attention to that. And we hope that, indeed, governments will pay attention. And unfortunately, depending on the government, people do not pay attention. And I'm not going to say which government or which person, but we need to be aware of that. Because we have people that deny science and deny the impact of climate change on society. We all know about COVID-19, so I don't think we need to go over that. But COVID-19 should have taught us some new behaviors. Unfortunately, in my opinion, it has not, because global governance is not working properly regarding equity and regarding how do we share vaccines, for instance. Remember, we had vaccines in some countries and total absence of vaccines in others. So globally, we are not equal. That's something also that we need to think about. How do we have a governance in that aspect? And I'm coming towards the end. That's something also that really strikes me, and science has a lot to do with that. We need to start talking about mental health in a more global way. The modern life, the way we are doing things, has indeed increased the episodes in mental health disorders. Social isolation proved that during the COVID, and the lack of access to mental health, because still there is a stigma, globally speaking, with the disease. And that's something, that data really is, it was from WHO 2019. I tried to check for a new one, but not from WHO, so were just papers, so I decided to keep that one. The suicide rate among adolescents, and it was mentioned the importance of the youth is increasing. And it's the fourth leading cause of death among youth from 15 to 29 years of age. That's preoccupying. That really, and it's from WHO. And we need to remember that there are lots of people without access to adequate mental health care. And the epidemiological studies in Europe, United States, and Brazil estimate that the gap in access to mental health service is between 40 to 50% for adults and adolescents. And it has a tendency to increase also with aging. So you need to pay attention to that. Stigma, I read, doctor. And I think in here, it's not only a problem of science or governance, the community, everybody together have to pay attention to this. Technological advancements, I already said something in the beginning. Telemedicine will help, artificial intelligence, precision medicine, but this are going to introduce inequalities, because precision medicine is going to be available for a few, not for all. And the costs of health are going to increase. We're doing much better. We're producing new medicines with the biotherapies that are fantastic, but they are not affordable, as well as some of the vaccines. So we need to pay attention to this if we're planning on indeed having the 17 goals. In conclusion, challenges facing health and social welfare are complex and multifaceted. Technological advancements, demographic change, socio-economic inequalities, and environmental factors will contribute to provide either equitably or not so much equitable conditions. And governments, healthcare providers, and community must work together in order to provide a resilient system capable of standing future challenges. We need to have prioritized, but it's easy to say, but I don't know how to do it. That's indeed true. Equity, sustainability, and adaptability. Adaptability is easier, I would say, but equity is going to turn out to be something really hard to achieve, unfortunately, if we continue to move in the way we are doing nowadays. And here, are some reference, and I want to give special thanks to EurASc, European Academy of Science, to Academia das Ciências de Lisboa, and to, oops, doesn't want to come? Okay, it was two pictures. One was from my alma mater, Escola Paulista de Medicina, Federal University of São Paulo, and Academia Brasileira de Ciências. And thank you so much for inviting me for this talk. Thank you.
Okay, now moving to the second speaker, who is Daniel Sherman. Professor Sherman is a molecular biologist and neuropharmacologist. He is also the head of our division of medicine and life sciences. So he has been the director of the Foundation for Rare Genetic Disease since 2017. And his main research focuses on gene therapy, for instance, the quantification of dopaminergic neuronal death in Parkinson's disease. So is very important topics, and today Professor Sherman will speak about biological sciences for sustainability. So please, Daniel.
Thank you very much, and I would like to thank Professor Cardoso and Pombero for their hospitality in this palace, fantastic place. And thank you, Rodrigo and Sarah and Luis for the great organization of this meeting. So today I'm not going to present my research because I had the feeling, and this is what happened, that Professor Nader would present the health side of sustainability, and I am more going to present some other domain, which is how plant science, green science can help for sustainability. So when we want to address the topic of biological sciences for sustainability, we can address this on two aspects. One is transformed biomaterials, and I'm going to present two aspects of how we can transform biomaterials for sustainability, and sustainable health. For lack of time, I will not go into present that, but just give some hint of where we can move toward sustainable health by the use of biological technique. So, first, I would like to present you some aspect of biomaterials, which are cyclodextrins. So cyclodextrins are some kind of basket. You see here the structure, and so they are circularized molecule, and in the center of the basket you have hydrophobicity, and the outside you have hydrophilicity. So this means that this basket will be soluble in water, and inside, in the hole of this basket, you can insert hydrophobic molecules. So it's a good tool to solubilize in water hydrophobic molecule, and this is very useful for many applications. So you see we have three different types of cyclodextrin, alpha, beta, gamma, depending on the number of sugar, because this is a cyclic molecule made of sugar unit, 6, 7, or 8, and depending on the number of sugars, the size will increase. So how do we obtain this cyclodextrin? We obtain them from starch. So what is starch? Starch is a white granular organic polymer produced by all green plant. It's a polysaccharide comprised of glucose monomer joined by 1 to 4 linkage. And starch, normally, flour, is insoluble in cold water, alcohol, or other solvent. So in nature, you find it either as a linear polymer, and then the name is amylose, or as a branch polymer, and this is what we call amylopectin. It's made in the green leaves of plant. It's some kind of energy storage. You find it in chloroplast, which is the energy, the material, which take up energy from the sun in plant. And it is stored in chloroplast for plant to survive during the night. But also you find it in large storage, and you find it in roots, in tubercle. So below you can find it in potatoes, for instance. This is where you find most of the starch which is used by human beings, such as wheat or potatoes or rice. So how can we produce cyclodextrin out of starch? Simply by using enzyme, and this is where biology enters into the game, because this enzyme, cyclodextrin glycosyl transferase, is the enzyme which is cutting starch and making the circle. And here comes all the biological tools that we have in hand in terms of improving the capacity, efficiency, speed, resistance of this cyclodextrin glycosyl transferase. And for instance, just to give you a hint, by genetic manipulation, we have obtained thermostable cyclodextrin glycosyl transferase. So it means that we can now put this starch in some kind of cooking machine at 70 degrees, and the enzyme is doing cyclodextrin at a very fast rate, much faster than normal enzyme, because it is thermostable. So this is one application of what biology can do for sustainability, which means lowering the cost of production, inducing high amount of production of cyclodextrin. And for instance, one application of cyclodextrin is that in the hydrophobic hole in the basket, there is some affinity for cholesterol. So if you want to produce low cholesterol food, you just mix the food with the cyclodextrin. Cyclodextrin will take up cholesterol, and then you will produce low cholesterol cheese or low cholesterol milk, for instance. But the application of cyclodextrin are much, much larger than low cholesterol food. And this is, for instance, a book. You can have it on internet. It's an open access, where you can see all the different applications. I'm not going to detail them, but for textiles, cosmetics, separation, catalysis, supramolecular chemistry, and so on, you can use cyclodextrin. There is a huge amount of application you can make with this kind of product. Now I'm going to shift gear to another topic, is how biology, and more precisely how genome editing can be employed to solve some of the problems that Hélène, Professor Nader quoted. How can we use genetic manipulation to tackle the problems that you have presented? And thank you for introducing the urgency of what I'm going to do now, present now. So how can we increase sustainability by using genetic manipulation? So for this, many tools have been developed, like transfer of genetic material, like induced mutation and selection of, for instance, material with a better capacity of advantages. What I'm going to present is the last discovery, which is a real revolution now in the last 10 years in biology, which is called genome editing or genome priming and so on. So it's all based on the structure of DNA. And you see that in biology we have this recognition system, which is the genetic code, which means that by hydrogen bonding, the thymine base will bind to the adenine. So in front of a T, you will have an A, or in front of cytosine, you will have a G. So there is an alphabet here. And on the right side of the picture, you see that there is what we call Watson-Crick hybridization, which means that in front of this alphabet, on the right you have T, A, G, and G, and on the left you have A, T, C, G. C, G, yes, in front of C, sorry, blue is C. So, you know, if you have one series of letters of one side, you can exquisitely match the other side, and this is a recognition system. If you have more than 20 or 25 different letters, in the whole genome of a human being, only one gene, coding for one protein, will recognize this sentence of 25 letters. So this is an exquisite way to recognize a specific locus on the genome. And this is what is being used now by genome editing. But this was found by pure serendipity, actually, and this is a proof on how fundamental research can be useful. Fundamental research was interesting on how bacteria can fight against viruses. You might think, who cares about how bacteria fight against their own viruses? There is a big war between bacteria, and there are some viruses, like tobacco viruses, which is attacking bacteria and destroy bacteria by injecting its own DNA into the bacteria. And some people were interested on some bacteria can resist, and they develop a defense system, an immune system, which means once a bacteria has been infected by a virus, if the bacteria has survived, now it has a specific defense, and it will survive and it will resist to any new infection by the same viruses. This is what we call immunology. We thought it was only existing in us, as how we can fight viruses, how we can fight bacteria. No, bacteria, they have their own immune system. So how does it work? It works by, and here you see on the top left bacteria, and you have the DNA, circular bacteria, the chromosome of the bacteria. It works by a specific recognition system where you see on the top two lines, you have a little sequence. And this little sequence, which is in black and white, in fact, it's a sequence belonging to a virus. This means that this bacteria has been infected by the virus before, and it has taken a little piece of DNA, 25, of piece of DNA from the virus, and it has integrated into its own genome. So the CRISPR locus, it belongs to the bacteria, and you see the different, you see cross, bar, small line between the R region. All this belongs to different viruses. And once the bacteria has incorporated into its genome this little sequence that we call spacer, this makes the bacteria resistant to the virus. So this was an interesting discovery, but so what? How does it work? It works because or maybe I was a bit too fast. Yeah, here. It works because the bacteria has a system which is the blue thing here. It's called CRISPR RNA, the transactivating CRISPR RNA, the track RNA. And this blue thing is binding to the R region, which is common everywhere. And it is cutting with the red arrow. And then it takes with it below, at the bottom, you can see, I don't know if I can have a pointer, it doesn't work. You see below you have this track RNA, and it was taken by the R, because it is binding by Watson-Crick hybridization to the R region. And the orange spacer is a little piece of virus. It's a little piece of DNA of the virus. So now there is this equipment here, which this track RNA and the yellow, which is the recognition system on bacterial DNA, and the orange, which is a spacer belonging to a virus, this will do the following. This will associate to a protein whose name is Cas9. And it is represented here. you see, it will bind to Cas9. Actually, you don't see Cas9. I don't know why. The Cas9 has disappeared. It's some kind of gray big protein. And it enter with this orange sequence here. It will enter into the DNA of the virus, and it will destroy, it will cut the DNA of the virus. So this means that if you have this Cas9 system, the bacteria is going to be able to recognize the DNA of the virus and to destroy and to resist to the infection. So this was extraordinary. Bacteria, they have an immune system. And in a few years, a matter of 2 or 3 years, it was demonstrated that you can use this machinery, the Cas9 protein, and this blue RNA, to enter into the DNA of our own cell, or plant cell, and to destroy specifically at a very specific spot some piece of DNA. So you can target out of 2 billion base only one sequence of about 20 base that you are targeting, and you want to act on this sequence on genomic DNA, not only in bacteria but also in plant or in human. This is the basis of genome editing. So after that, we what we did was to make only one, because you see this blue, yellow, and orange is a little bit complicated. You have 2 things, and you have one. The blue is linked by Watson Crick to the yellow and orange. So the idea was to make a single one and only one molecule. And this is what we get here. I'm sorry, the Cas9 has disappeared. I don't know why, a problem with the computer, but no problem. The protein Cas9 is there. And you see, now you have this single guide RNA, we call it LG-RNA, which is recognize the sequence. And this is also for any kind of human or plant cells. So how can we deliver that? This Cas protein and single guide RNA. So you can imagine there is a large gray protein around this system. And how can we deliver this system to the cells? So again, you have to imagine that you have the gray protein inside. He's the only one who is able to see my presentation, actually. I don't know what happens. Anyway. So it is inside this particle that we call lipid nanoparticle, which is another evolution, because these lipid nanoparticles are made of, you see below, cationic lipid with a PEG polyethylene glycol, cholesterol, and neutral lipid. And these lipid nanoparticles are the ones that have been used for the mRNA vaccine against COVID-19. So this is another evolution. I have no time to detail that. But this is what we use to deliver mRNA vaccine and to make vaccine against COVID-19. At least the new mRNA vaccine by Moderna and Pfizer. Okay. But here what we use them to deliver the single guide RNA of CRISPR and the Cas9 protein. The whole thing is inside the lipid nanoparticle. And this is one example of what happens when you deliver it in vivo. You deliver it into the blood of a patient, and then it will bind to the ApoE, apolipoprotein E, because PEG polyethylene glycol has an affinity for ApoE. And then on the top right, you will see this will bind to the ApoE receptor, the protein ApoE receptor on the membrane of liver cells. And then this will enter into the cell, and it will deliver to the endosome, and then into the cytosol, and then into the nucleus. So now this is the beginning of my presentation. It's a genetic improvement of plant for biosustainability. So there is urgency, but I mean, I don't need to comment that. Hélène, you have presented that enough. So genetic engineering, for instance, in trees, what can we do with that? We can we can obtain, and why do we do? And what can we obtain? we can be active and positive effect on economic development, on conservation and sustainable use of genetic biodiversity. We can also provide large-scale forest restoration, reduction of air pollution, acceleration of biomass production, bioenergy, and inducing flowering to improve the production of fruits by trees. This is a large scope of what we can obtain by genetic improving plants by the tools that I have presented. So, for instance, to give you, for example, we have genetically modified eucalyptus for accelerated growth and increased biomass accumulation. There has been crops, cotton, maize, soybean, which have been engineered. And here we have inserted the Bt gene, the Bacillus thuringiensis gene, which induces that we don't need we need less insecticide and less pesticide for production. And for instance, another example is tomato, gene-based development to improve the quality of tomato, to induce higher firmness, also shelf life. New genetically induced tomato, you can have shelf life for weeks, even months, and protection against stress. So you see there is a large amount of science, and this is another book that was published addressing the UN Sustainable Development Goals of woody plant through genetic engineering. Just, I want to present biofuels, okay? I have no time to comment, but of course there is some genetic modification, for instance, how to degrade lignin. Lignin is a family of polyphenol macromolecules that are an obstacle to obtain carbohydrate for biofuel. So by genetic manipulation we can improve decrease the amount of lignin, and we can also, for instance, improve some enzyme. You have this single guide RNA, we call it LG-RNA, which is recognize the sequence. And this is also for any kind of human or plant cells. So how can we deliver that, this Cas protein and single guide RNA? I think your computer doesn't like the gray color, or do you see the protein? You see it on the screen, but I don't see it, and it doesn't come. So there is some kind of barrier here. Anyway, so you can imagine there is you have this single guide RNA, we call it LG-RNA, which is recognize the sequence. And this is also for any kind of human or plant cells. So how can we deliver that, this Cas protein and single guide RNA? I think your computer doesn't like the gray color, or do you see the protein? You see it on the screen, but I don't see it, and it doesn't come. So there is some kind of barrier here. Anyway, so you can imagine there is a large gray protein around this system, and how can we deliver this system to the cells? Okay. So again, you have to imagine that you have the gray protein inside. He's the only one who is able to see my presentation, actually. I don't know what happens. Anyway, so it is inside this particle that we call lipid nanoparticle, which is another evolution, because these lipid nanoparticles are made of, you see below, cationic lipid with a PEG polyethylene glycol, cholesterol and neutral lipid. And this lipid nanoparticle, you have this single guide RNA, we call it LG-RNA, which is recognize the sequence. And this is also for any kind of human or plant cells. So how can we deliver that, this Cas protein and single guide RNA? I think you have this single guide RNA, we call it LG-RNA, which is recognize the sequence. And this is also for any kind of human or plant cells. So how can we deliver that, this Cas protein and single guide RNA? I think your computer doesn't like the gray color. Or do you see the protein? You see it on the screen, but I don't see it, and it doesn't come. So there is some kind of barrier here. You have this single guide RNA, we call it LgRNA, which is recognize the sequence. And this is also for any kind of human or plant cells. So how can we deliver that, this Cas protein and single guide RNA? I think your computer doesn't like the gray color. Or do you see the protein? You see it on the screen, but I don't see it, and it doesn't come. So there is some kind of barrier here. Anyway, so you can imagine there is a large gray protein around this system. And how can we deliver this system to the cells? You have this single guide RNA, we call it LgRNA, which is recognize the sequence. And this is also for any kind of human or plant cells. So how can we deliver that, this Cas protein and single guide RNA? I think your computer doesn't like the gray color. Or do you see the protein? You see it on the screen, but I don't see it, and it doesn't. You have this single guide RNA, we call it LgRNA, which is recognize the sequence. And this is also for any kind of human or plant cells. So how can we deliver that, this Cas protein and single guide RNA? I think your computer doesn't like the gray color. Or do you see the protein? You see it on the screen, but I don't see it, and it doesn't come. So there is some kind of barrier here. Anyway, so you can imagine there is a large gray protein around this system. And how can we deliver this system to the cells? Okay. So again, you have to imagine that you have the gray protein inside. He's the only one who is able to see my presentation, actually. I don't know what happened. Anyway, so it is inside this particle that we call lipid nanoparticle, which is another evolution, because these lipid nanoparticles are made of, you see below, cationic lipid with a PEG polyethylene glycol, cholesterol and neutral lipid. And these lipid nanoparticles are the ones that have been used for the mRNA vaccine against COVID-19. So this is another evolution. I have no time to detail that. But this is what we use to deliver mRNA vaccine and to make vaccine against COVID-19. At least the new mRNA vaccine by Moderna and Pfizer. Okay. But here what we use them to deliver the single guide RNA of CRISPR and the Cas9 protein. The whole thing is inside the lipid nanoparticle, and this is one example what happens when you deliver it in vivo. You deliver it into the blood of a patient, and then it will bind to the ApoE, apolipoprotein E, because PEG polyethylene glycol has an affinity for ApoE. And then on the top right, you will see this will bind to the ApoE receptor, the protein ApoE receptor on the membrane of liver cells. And then this will enter into the cell, and it will deliver to the endosome, and then into the cytosol, and then into the nucleus. You are standing because I am at the end of my presentation? 5 minutes, yes? Oh my God, no. Okay, so now this is the beginning of my presentation. It's a genetic improvement of plant for biosustainability. So there is urgency, but I mean, I don't need to comment that. Hélène, you have presented that enough. So genetic engineering, for instance, in trees, what can we do with that? We can, we can obtain, and why do we do? And what can we obtain? We can be active and positive effect on economic development, on conservation and sustainable use of genetic biodiversity. We can also provide large-scale forest restoration, reduction of air pollution, acceleration biomass production, bioenergy, and inducing flowering to improve the production of fruits by trees. This is a large scope of what we can obtain by genetic improving plants by the tools that I have presented. So, for instance, to give you, for example, we have genetically modified eucalyptus for accelerated growth and increased biomass accumulation. There has been crops, cotton, maize, soybean, which have been engineered. And here we have inserted the BT gene, the Bacillus thuringiensis gene, which induces that we don't need we need less insecticide and less pesticide for production. And for instance, another example is tomato, gene-based development to improve the quality of tomato, to induce higher firmness, also shelf life. New genetically induced tomato, you can have shelf life for weeks, even months, and protection against stress. So you see there is a large amount of science, and this is another book that was published addressing the UN Sustainable Development Goals of woody plant through genetic engineering. And just I want to present biofuels, okay? I have no time to comment, but of course there is some genetic modification, for instance, how to degrade lignin. Lignin is a family of polyphenol macromolecules that are an obstacle to obtain carbohydrate for biofuel. So by genetic manipulation, we can improve decrease the amount of lignin, and we can also, for instance, improve some enzyme. Oh, you can decount this time from my time. You know, okay? Okay, so now, okay, sorry. So now I want to present this by my colleague Kik-Chuan Tong from EurASc, which was elected in our division 2 years ago. And I will you have this single guide RNA, we call it LG-RNA, which is recognize the sequence. And this is also for any kind of human or plant cells. So how can we deliver that, this Cas protein and single guide RNA? I think your computer doesn't like the gray color, or do you see the protein? You see it on the screen, but I don't see it, and it doesn't come. So there is some kind of barrier here. Anyway, so you can imagine there is a large gray protein around this system. And how can we deliver this system to the cells? Oh. Okay. So again, you have to imagine that you have the gray protein inside. He's the only one who is able to see my presentation, actually. I don't know what happens. Anyway, so it is inside this particle that we call lipid nanoparticle, which is another evolution, because these lipid nanoparticles are made of, you see below, cationic lipid with a PEG polyethylene glycol, cholesterol and neutral lipid, and these lipid nanoparticles are the ones that have been used for the mRNA vaccine against COVID-19. So this is another evolution. I have no time to detail that, but this is what we use to deliver mRNA vaccine and to make vaccine against COVID-19. At least the new mRNA vaccine by Moderna and Pfizer. Okay, but here what we use them to deliver the single guide RNA of CRISPR and the Cas9 protein. The whole thing is inside the lipid nanoparticle, and this is
tolerate temperature, they tolerate contamination, they tolerate different substrates. We use residues and waste as substrate, convert them via insects into protein-rich feed, which can then hopefully one day replace a very unsustainable fish meal production globally, which is currently 5 million tons per year. I won't touch the insects today. I want to focus with you today a bit on the volcano crater algae, and this is called Galdera sulfuraria. What is so interesting on this algae? First of all, it stands for short before novel food approval. So this is not just a dream. This is just short becoming reality from my point of view. The algae can produce up to 64% protein in the biomass. It can run at pH 0. I was really impressed when our team lead in biotechnology, Dr. Fabian Abusi, came to us and suggested this solution. We can run a continuous cultivation at pH 1, normally we do that, and without decontamination. This is the biggest problem in fermentation, if you want to run it over weeks and months, that you have decontamination, or that you have contamination and you need to decontaminate. It's quite thermotolerant, so we don't need to cool or heat in subtropical and tropical areas. Cooling specifically in these areas is very energetic and costs money. We have a stable continuous cultivation demonstrated in ETH Zurich over 3 months, and we could run over 50 different residue substrates, also in Singapore now with our microalgae, to replace commercial media, which is really a strong cost driver and always limiting your sustainability performance. We can run it with photosynthesis, and then you would have a green microalgae, and that is the most known microalgae, or we can run it in the dark, in heterotrophic conditions, and that would be a white or yellow microalgae. You just need to change a bit the substrate composition. Or you run it in between, and that is called mixotrophic cultivation. Here our team lead Dr. Abusi developed a mixotrophic oxygen CO2 balance cultivation, so we don't need to inject any gas. And this is a very interesting aspect because even if we say we take the CO2 from the atmosphere, very often we run our microalgae with tanks next to it and inject the CO2, for example. Here we now don't need to inject any O2 or CO2. That is enabling a simplification of the process, making it more robust, flexible, and that is reducing capital expenditures, operational expenditures, and hopefully it's making it more affordable. This needs to be still demonstrated. Regarding regulatory, in the US, you can run such a system with self-affirmed, general recognized as safe, GRAS status. In the EU, you can see here the novel food dossier, which is currently under evaluation, and we expect an approval hopefully soon because it runs already since several years. Yeah, but what is then the right key performance indicator? What is the right production? Is it the protein concentration? Is it the productivity? Is it maybe even more nutritional qualities like bioaccessibility? Can we digest the material? Yeah, that is a good question. Here we could demonstrate the different productivities of autotrophic and mixotrophic growth, where we could show with mixotrophic cultivation a 2.3 times higher productivity, much more biomass. With both strains of the same species, Galdera sulfuraria, we could produce protein-rich biomass, 64%. You could say, okay, let's go for 64. But then we did an in vitro digestion analysis and could demonstrate that only 14% of the protein was digestible, bioaccessible. We used the standard protocol InfoGest 2.0 from the European ecosystem. And if we would accept maybe a bit lower protein concentration, we could show that we could have a 62% bioaccessibility, and then I would suggest let's maybe go for a bit less protein in the biomass and let's go for more bioaccessibility. And then we come now to the question, is it bioavailable? Can it be absorbed in our intestinal tract to the blood? And is it bioactive? That's all big questions of many novel foods which we still need to answer. On top, we could demonstrate that we have a lot of high concentrations of iron and zinc. Iron and zinc are one of the most important micronutrients which are very often linked to deficiencies, not only in developing areas, also in the developed world. Yeah, so what can we do to make that more affordable? I already touched that a bit. If we have a high quality protein-rich biomass, we can justify easier CapEx and OpEx cost. If we have a flexible cultivation, we can adapt it to regional needs. It's not everywhere the same conditions, so we need to have a solution which is flexible enough, and I think this would be the case here. As we have more than 50 residues as substrate and media demonstrated, again, this flexibility gives us certain advantages. However, if we take residues, we need to add a hydrolysis line into our calculation, into the investment, but also into the running cost, because hydrolysis very often, if we do it with thermal hydrolysis, takes energy. We can grow it at pH 1, and that is really a significant game changer in terms of investment into the reactor. We can have a simplified reactor. We don't need to work with sterile conditions. We don't need to decontaminate in our operational cost, significantly reducing efforts here. And we can run it with higher temperatures. And that is, again, avoiding cooling needs very often if we run it in tropical or subtropical areas, which are very often using too much energy. And last but not least, I mentioned to you the oxygen-CO2 balance cultivation, which enables us to avoid any gas injection. So I think this equation can be quite successful. Now we need to prove it. We currently run a 50-liter reactor system, also with industry partners and startups, to demonstrate the feasibility. On top, we could develop a technology which is called pulse electric field cell stimulation. This technology is already used in the industry for potato processing, and we use it now for single cells. On the left, you can see that we could increase cell biomass up to 40%. It grows just faster, not more, but with time, you save money and efforts. And this technology was, you can see the little picture, commercialized now with my first doctoral student from ETH Zurich and the Bühler company in Switzerland. Now they run it up to 20 tons per hour. On the right, you can see we can also use this technology if we go to microsecond domain to open cells slightly and milk them, keeping them alive. Maybe that is enabling new approaches in continuous cultivation. Why we need to always use the same approach from plants in fermentation to grow it and then kill it and then harvest it. Maybe we can run it in a continuous mode and have a continuous milking of nutrients of live cells. And last but not least, we can also have a gentle disintegration, non-thermal, non-chemical with this technique. On top, we can also use then this protein-rich biomass with another emerging technology, which is called high moisture extrusion. That is one key and enabling technology to produce meat substitutes to structurize material. Without structurization, you don't have success on the market. So here we take protein concentrates from plants, from soy in this case, which is the best protein from the plant side, and we added yellow microalgae, heterotrophic cultivated microalgae protein concentrate to demonstrate the feasibility producing structurized products. And we could do that up to 50%. You can see the little picture here of a, let's say, yellow-like meat substitute, which is a bit copying the structure of a chicken meat. On top, we could increase the micronutrient concentration, vitamin E, B1, B2, B3, and B6, even after extrusion. And that is, again, meeting the need for nutritionals meat substitute. Very often, our protein-rich alternatives don't have the same nutrition than the animal origin. And if we want to reduce animal-based foods, we need to offer the same nutrition from my point of view. Yeah, but this is just the beginning, because we talk here about concentration, digestion, but is it bioavailable? Very often, nutrients in plant-based meat substitutes, like iron and zinc, are not bioavailable because they are blocked by inhibitors in the intestinal tract. So we need to prove how we can increase that to, again, meet the same nutritional quality of the animal origin. And we do that here with the bioaccessibility in vitro studies and optimization of our bioprocess, but also bioavailability testing ex vivo with KQ2 cells and in vivo with mice and humans, where we hopefully can demonstrate then the different benefits of novel foods, but also of existing protein-rich food alternatives. We do that with the ETH nutrition research of Professor Ferdinand van Nijen. And with that, I hope I could explain to you if the tracker is working. No, it's not. What we understand from sustainable food processing, so we do food process development in different domains, but also a parallel multi-indicator sustainability assessment. I think this is really needed if we talk about sustainability, that we are quantifying sustainability performance in all three dimensions. Methodology exists, even ISO-normed, like life cycle assessment, that identifies opportunities and trade-offs. We can then tackle our solutions in the right sweet spot, where we focus in our team on nutritious, affordable, and more sustainable protein-rich foods to hopefully reduce the number of animal-based products. But these must be affordable, otherwise they don't have any impact globally. Here we need to reduce capital expenditures, operational expenditures, And I hope I could demonstrate to you that polyextremophilic organisms from the biotech side can hopefully here bring some interesting aspects into the solution space. But I would like to emphasize we need to have a fair comparison of these new solutions regarding innovation readiness level to the benchmark and highly scaled one, that we can really run that in a successful mode over the innovation cycle. And last but not least, let's do again the sustainability assessment that we really are sure that we go into the right direction, and that needs to include all the 3 dimensions. With that, I would like to thank my team in Zurich and Singapore at the ETH Zurich and Singapore ETH Center. I would like to thank all our colleagues from the Zurich ecosystem, food research ecosystem, the research centers, the spin-offs, the startups, but also our industry partners, which support our scaling and technology readiness level increase. And I would like to thank you for your attention. Thank you.
Thank you so much, Professor Martin, for your lecture and given very interesting ways to have a better sustainable world by growing microalgae and eating insects. Why not? So there is just one question. Yes, by Daniel Sherman.
Very interesting talk, thank you. How do you compare your production of biomeat with culture cells? Because you know, meat is artificial meat now is improving in terms of production of cells in individual muscle cells. So how do you compare your technique to this one?
Yeah, thank you very much for this question. We do a sustainability assessment, actually, where we quantify the environmental, economic, and social impacts as best as we can. Very often, with cultured meat, we don't have enough data. That is an early solution. But what I see here for as improvement for cultured meat, definitely the media cost need to go down. And currently, we need too much purified media. The conditions to run it, very often sterile conditions or using antibiotics, it's not beneficial for food production, but now there are some more solutions, but we don't get access to the data, so it's very hard to compare. In general, I think plant-based foods are very hard to beat, even with microalgae so far in terms of affordability on the market. Structured and as a real meat, let's say, the cultured meat can bring interesting solutions because it's a pure solution. And in Singapore, I observed it, of course, in our laboratory as well.
Can you give your more detail on your electroporation system?
Yeah.
How does it work?
Yeah, electroporation is not something new. It was discovered in the 70s, you know most probably best, to bring in out RNA and DNA. We use it now to soften potatoes up to 100 tons per hour globally in China, in North America and Europe. Now we use this technique to stimulate cells in nanosecond domain to grow them faster. How it works, we stress the cell. It's an internal stress response.
Okay.
Okay, thanks.